Tim, thank you very much for this wonderful presentation. I really enjoyed it very much, and I really enjoyed the part when you mentioned that not only success is actually coming during scientific activity, but also wrong experiments or unsuccessful experiments happening. If you would calculate about how many experiments you would quote as successful ones and how many experiments you would quote as unsuccessful one, how would you put this rate about in your career?
Well, that's a difficult one to answer, and you know, you sort of tend to remember the good times and forget the pain and the bad times in between. But you know, I mean, real successes come maybe every 10 years, and in between it's mostly troubleshooting because things aren't working properly. But of course you have sort of you have to break it down, and I used to like going to the lab and seeing whether last night's experiments had worked, you know, and if they did, you felt a little bit better, and on you would go. But I think the ratio is really very low indeed of real success. Very low. I mean, you know, 1% or maybe even lower.
Yeah, so people shouldn't be actually unmotivated due to the plenty of failure during their experiments.
No, you have to know what you're looking for. That you have to have courage and faith that eventually you'll break through to the end. But often it's it's really quite depressing, and very often too you don't really know where the failure, you know, what's the origin of failure? Is it because your ideas are completely wrong, or because you're technically incompetent, or because somebody in the lab is not pulling their weight? I mean, it can be all kinds of things. Very difficult. Very difficult.
So actually a question coming from the students, from, for example, Bibor Mate, who actually asked that what was really your motivation after your failures at the beginning? So what was your driving force?
Well, I think there are two things. One is, in my case, I really don't know what else to do. So you only have one possibility, which is working in the lab. But above all, I suppose, is, you know, an underlying self-confidence and an underlying curiosity. I would say curiosity is the main thing, you know, sort of real burning desire to understand something. But it's finding that something to understand which is perhaps the most difficult thing in my experience, because of course we would all love to know how the brain worked, how you think, but that's a bad problem because it's very poorly defined. You don't know how to tackle it. So it's the key, I think, to, at least as I see it, for successful research is to find something which you think you can solve in a reasonable time with the resources at hand, and then you just have to struggle to figure out how to get that. But a good problem is one that takes perhaps, you know, several years to solve, but is an interesting problem that other people are, you know, competition is also important. I think it's good to have friends and competitors who are interested in the same thing, because you can share the successes and share the failures and discuss things freely and frankly with them. I'm a great believer in openness. Some people get very paranoid and think that, you know, they're about to get scooped or stuff, but I never sort of worried too much about that.
Yeah, actually, and with the same topic, another students from Daniel Vidach coming the questions, but what do you think about the process of troubleshooting? What is the most healthy way of doing it, and what is the most efficient? Is there a common nominator or decision making between the two ways?
Yeah, I don't know. Some people are very good at it, and you know, and often it's very trivial things. I remember a time I moved from Cambridge to a cancer research lab, which is just outside London, and we used to make all our own enzymes. And because the second lab was much better funded, we decided just to buy these enzymes which had been making in the past. So we switched from the homemade supply to a commercial, very reliable commercial supply. And the reaction stopped working. And the graduate student in charge of this particular project took a very long time to figure out what was wrong, because she trusted so much the commercial enzymes that the one thing she didn't test was whether the new enzyme which we bought was as good as the enzyme we made. And it turned out in the end that it wasn't, and for a very simple reason, which was that they simply hadn't mixed the solutions up properly. You know? So what they'd sold as T7 RNA polymerase, which is actually 90% bovine serum albumin with a tiny, tiny trace of T7 RNA polymerase, whereas our stuff was 100% T7 RNA polymerase. And she took a long time to figure that out. And that was very interesting. She should have figured it out in less than a week, and she actually took about a month. She was terrific. I mean, she was a wonderful graduate student, but that was sort of indicative, actually.
Yeah,
so little things like that.
Yeah, it's never easy, and finally to find a way why your experiments don't work, especially making differences between the biological and non-biological.
Yeah, I mean, another wonderful example. We had an American student in the lab one time, and his reactions kept on drying out. You had to, at some point, put them in an oven, and his always dried out. We said, Ted, you know, you just have to cover them with saran wrap. He said, I do. And it turned out that our lab had two kinds of this cling film in it, one of which was proper saran wrap, which was very impermeable to water, and another one, which came from my local supermarket, which, for reasons I don't understand, was permeable. And Ted had been using the wrong one. It took us ages to figure out that he was putting saran wrap, but the wrong kind of saran wrap over his tube. I may say he's gone on to be a very successful neurobiologist, so it didn't seriously damage it. But it was very telling that, you know, I mean, really teeny weeny trivial things like that can set you back for weeks.
Sure.
So watch out. And in fact, I mentioned that my very first research project didn't work, and I didn't tell you what the reason for that was. The reason was I hadn't read the materials and methods of the paper carefully enough, being very inexperienced. And I'd used the wrong kind of rat. You know, we had one kind of rat, and the kind of rats I should have used was some other different special kind of rat. But you know, I think that was lucky in my case. I didn't read the procedures properly, carefully enough. I mean, it's like, I don't know, I'm a very keen amateur cook, and reading recipes is rather like that, you know, and sometimes the soufflés rise and sometimes they don't. And well, I know you have, Peter, you live in a wonderful cook's family. You know all about this kind of thing. You know, there are recipes and there are recipes, and sometimes they work and sometimes they don't, and sometimes you understand why and sometimes you don't. It's very simple. Yeah. I mean, all life is like that, really.
Yeah, this is the most exciting issue in science, to find them out. Actually, two questions coming from two different students, but both of them connected with each other. The one question is coming that why you choose cell division or as a research topic to go on, and the other question is coming that in your career, when you realize that your discoveries may worth the Nobel Prize one day.
Well, as far as cell division was concerned, I tried to explain that in my talk. I mean, that I was really an expert in the control of protein synthesis, but because of switching to sea urchins and clams, we began to realize that that after fertilization, the most important thing that these cells did was to divide. We didn't really fully appreciate that division was rather special, because unlike most cells, when they divide, they have to grow. Eggs, when they divide, are simply subdividing themselves so that they don't have to grow. They don't have to make they really don't, in principle, have to make anything new except DNA. So why did they have to make new proteins? And people simply hadn't focused on that. So I got sucked into the business of the control of protein synthesis. And then when I discovered that a protein was degraded in order for the cells to divide, I mean, that was an extraordinary thing. And that's a very good example, I think, of seeing by accident something that people, in fact, in this case, had not even theoretically proposed. It was so impossible that cells could destroy one protein at a particular time in the cell cycle that it was ridiculous. I mean, that was unthinkable. So I stumbled on something which was totally unthinkable. And that also answers the second question. That's why it's a Nobel Prize winning experiment, because I discovered something that people thought was impossible. But because I saw it with my own two eyes, it just, you had to deal with it. And there was no getting away from it. So I immediately realized, actually, I wrote a letter home saying, I've just stumbled on something. I've discovered something which I think is probably the most important discovery of my life. And so it was. I mean, I'm just rather lucky that the Nobel people think that kind of discovery, especially in medicine, that sort of totally unexpected discovery, is exactly the thing they give prizes for. They don't give prizes on the whole for people who are really good. I mean, I'm not a brilliant scientist by any manner of means. I've met lots of scientists who are much cleverer and more able than I am, but I just happen to have a knack for noticing interesting new things.
Yes, you always quote that you like being more cleverer people with you, but the others, I think, exactly think the same when they are chatting and discussing the results with you. So I think you are definitely not only lucky in this part, but also your hard work, curiosity, and all other were necessary for all of your success. Actually, very interesting question coming from students, and it's always a good question is about your mood, whether in your career, how would you consider that the funny mood at work or good mood at work would have to be successful? So work success and about your mood or inner motivation, how important is, or maybe your commitment to your questions is the most important part.
Well, I think the mood is very important, actually, and happy, well-regulated labs help enormously with scientific progress. Again, I can remember a time when, for reasons that I'd never really fathomed, we had, for the most part, a very happy lab, and I liked to hire people who I liked, and I occasionally could see, in the early days, I had a lot of undergraduates passing through the lab, and they did projects, and I used to encourage people to go off to places which I thought would suit them as far as possible. I mean, I didn't try and keep all the very clever ones. I tried to keep the ones that I thought I could work with me, and that was important. And the result was that one had a cadre of people who liked each other, respected each other, and who could work together as a team. I mean, you know, Peter, very well all about football, how important it is that people get on well together, they respect each other's strengths and weaknesses, know how to help, and that sort of thing. I think that's tremendously important, actually. Perhaps the most important thing of all, especially in running a lab. I mean, it's teamwork.
Yeah, thank you. Another question coming from students maybe who try to deal with the work-life balance, that what do you think if you had to miss something in your life, actually to be able to spend your time with research and science? So at going on the long run in science, you miss something which you couldn't do because the time.
Well, I was very lucky actually. I mean, the time, most of the time when I spoke in that talk, I didn't have any sort of connections. I didn't have a family. I didn't have children, and so I devoted myself more or less full time to working in that was my life actually. The lab was my life, and even I didn't really take any vacations because I went to enough meetings, so lots of foreign travel was involved, so that was okay. So it was pretty sort of full on committed to the life of the lab, and that suited me at the time. Later on, I got a wife and children, and then I realized, you know, how important they were, and I couldn't then devote myself in the same kind of way to research. So I think it's very hard to make the balance. But having said that, you know, there are lots of different ways of doing research, and people are very successful about working out how to do it. And there's no doubt that there are as many different kinds of scientists as there are people on Earth, you know, and everybody has their own way of working. You just have to work it out for yourself, what you do.
Actually, another question coming, because of course the lifestyle is definitely different nowadays than maybe 30, 40 years before. So in the current world, in this fast-paced world, what would you do differently now than at the beginning of your career?
Well, I don't think I could do anything differently, actually. I don't know. That's an impossible question to, it's a totally hypothetical question, so I'm not going to try to answer it.
Okay, the another question was that you mentioned also in your presentation somebody who made a bad conclusion. So when you see your results, how can you make sure that the conclusion you try to make from the results is a good one or a bad one? So what can drive you through on this process?
Well, I think the most important thing is, I mean, you have to be extremely skeptical and self-critical, but therein it helps to have colleagues who you can discuss with, you know, and it also helps to have again, I go back to the thing of competition, people you're scared of, you know, they might get there first. So, you know, you have to really be able to convince them that you're right. And I think the most important thing really is to be your own harshest critic. I mean, try not to deceive yourself. And early on, I had one or two cases where I, you know, made a mistaken interpretation, and people corrected me, and I didn't like that. I didn't like people saying, "Oh, that's Tim, that's stupid." You know, you've fallen into an elementary trap. So once you've made that mistake a couple of times, you try very hard not to repeat it, and you become more self-critical. I mean, you don't want to be too self-critical. One of the problems often with beginning scientists is to say, "Oh, I'm not going to try that. I'm sure it won't work." I mean, the important thing is to do the experiment, and the important thing in my experience is to try and reduce the energy barrier to doing an experiment. You want experiments to be the easy thing to do, and you don't want to discourage yourself from doing experiments. So figuring out how to do that is quite tricky also, I think.
Yeah, actually another question coming, Vader, if you could ever imagine how big of a therapeutic breakthrough the detection of cyclins would be when you made your discoveries.
Well, you know, after discovering cyclins and realizing how important they were, I was sort of on a high for about a year, actually. I mean, making a really important scientific discovery is a tremendous boost to confidence. And the only trouble is that I also briefly alluded to in the talk was that when you've made a discovery, the problem is, where is the next one going to come from? it's a, you know, you sort of build up this discovery capital and then it dwindles and dwindles and dwindles away. And several times in my life I thought, oh, you know, I'm finished. I'm never going to find out anything ever again. And you just, under those circumstances, you just have to keep going. But it's tough. It's very tough. So it's, you know, it's a high and, and a low, and then a high again, and then a low again.
Sure, another question. Life. Yes. So right. Another question coming. I don't know whether you will or you can answer that. The question is that if you hadn't chosen science, or you couldn't have chosen science in your life, what other kind of career would you have imagined for yourself?
Well, I think from a very early age. I mean, I sort of discovered that I was good at biology. I was very lucky. I discovered that when I was about, I don't know, 11 or 12 years old. And I never seriously considered anything else, actually, because I wasn't very good at anything else. And I think one of the things that I always advise young people is try, you know, the sooner you can find out what you're good at, the better off you are. And in some ways, the most cursed people are the people who are extremely talented at everything because they can never decide what to do, right? Because anything they do is successful. I was lucky. I could only do one thing, which was science. So I never had any alternatives to distract me.
Okay, another question coming concerning the environment and other professionals. What do you think, how much affect in your work really the different environment, or if you have any other things to do? Rather these are distracting you, or rather you can get knowledge from the others and maybe can use another knowledge to your daily research?
Well, obviously the environment makes a difference, and I was very lucky in where I studied, both in school and university and in the various labs that I inhabited. And even now, here I am in Okinawa, which is a beautiful subtropical island, and you know, hardly a day passes that we discover something new. I mean, for example, this is a sort of silly thing that gives me pleasure. It turns out that there are a lot of mulberry trees in Okinawa. There are several on campus and there are several along the road. And you know, for 4 years we never noticed these mulberry trees. And mulberries are delicious fruit, actually. So they grow wild here. And how can it be that we missed them for 4 years? Was there always a bad year for mulberries, or were we just blind? So, you know, it's a weird thing. You don't always realize the advantages your environment provides for you. But I think the environment is important. I mean, it will affect what you do. It'll redirect you. But you have to keep your eyes open, and you have to look for opportunities too. You know, I'm a great believer in people grabbing their opportunities. You know, if you see a chance, go for it, because it may never come again.
Okay, another question is maybe an easy question, that have you ever considered giving up research during your career?
Oh yeah, quite often. Quite often. I mean, I have given up research now. When I was 67 years old, I retired from running a lab because I found running a lab very demanding, and I'd had some health issues. I had some heart trouble, and after that, it was much better. I felt much better after retiring, I must say. I'm not a natural leader. I find having responsibility very difficult. And when you're in a lab, you know, and you're in charge, as you know, you have a lot of responsibilities to other people, and that wasn't so great. I don't like bossing people around.
So actually, another question coming, that if you could only choose one person, a single person who inspired you the most, who would be that?
Well, I think early on, the person who inspired us most, but he was sort of unattainable goal, with Francis Crick was the inspiration for my generation, because he was just so smart, and he had a sort of underlying simplicity about his smartness, which was very, very attractive. You know, he always made things simpler, and that was great. Other people, you know, made things more complicated to try to prove how clever they were. He proved how clever he was by making things simple. And I think that's a great art. And almost all the really great scientists, I decided that, you know, I've known a lot of Nobel Prize winners in my time because I grew up in Cambridge in the great glory days of molecular biology. So I knew a lot of these great pioneers. And the extraordinary things were how different they were from one another. So, you know, did they have anything in common? And I think the things they had in common was that they liked to get to the basic underlying simplicity of things. When you're young, you like to make things very complicated because it sort of shows how clever and multifaceted you are, and you can put things together. But the real art of being a good scientist is to show that actually nature is fundamentally simple. But it looks very confusing initially, and just trying to tease out the underlying simplicities is what it's all about. Very easy to say, very hard to do in practice.
Yes, indeed. And actually, a last question from a student from this session is that what is your current goal in your life right now? So what is
my current goal? Well, my current goal actually is to be able to walk properly again, because I broke my leg just before Christmas. But I have one or two projects on the go. So one project is concerned with this.
Wow.
My friend John Wilson and I, here's John. These photographs were taken with us. We're both lying on the beach, actually, not dancing in the air. And we're trying to put these problems into a sort of, there's a new edition. The big book is Molecular Biology of the Cell, which is a very good book, which I've been involved with since 1985. And we're trying to put these problems onto a online computer-based multiple choice format. And so far, I think we've been working on this for 5 years. And we have about 1 more year to go to complete it. And I think we're on track. But it's, so I email John almost every day, and we sometimes have Zoom conferences. So that's one of the things that occupies me when I'm in my office. And otherwise, it's looking after the dog and looking after my wife, and that's pretty important too. Since I'm retired, I don't have to be anywhere anytime. So until I broke my leg, I did most of the housework. Now, unfortunately, I'm handicapped. So it's proving a little difficult at the moment.
So thank you very much indeed. We really, really greatly appreciate your time. Congratulations to your book. This is wonderful, and all of your activities. And thank you for sharing your time with our students. And we really do hope that after this pandemic, and when your health, your legs became better, to get the chance again to welcome you.
I hope. I'd love to come back to Hungary, I must say. I'm running out of paprika.
Yes, either ways, either we go to take paprikas to you, or you come to Szeged. Both ways will work.
And honey, that's the other thing which I'm running out of. Okinawa doesn't have good honey.
Okay. So